Okay, hi, we are Group 1, and we are this video is under BS 3336 of the Proteomics workshop, and we'll be discussing on acetylation today. So before moving on to the domain of acetylation, we have to first define the scope of this case study, which falls under proteomics research. So to set the baseline of this research, we have to first define what a proteome is. So the proteome is actually an entire set of protein expressed by the genome of a cell. So there is actually an increased complexity of human proteome to the human genome, as one gene may actually code for different multiple proteins. So to add on to this complexity, uh, another feature of the protein is actually the dynamic nature of it, which actually leads to differential responses to several mechanisms. So such uh, varied a plethora of structural and conformational change uh, is exemplified by one example, which is actually the post-translational modification, which actually is a common mechanism to regulate cellular activity. So this increases the functional diversity of the proteome. So to define what post-translational modification or PTM is the covalent modification of proteins which occurs after protein biosynthesis on the ribosome, which is after the translation. So under PTM, it falls under two categories, which is the enzymatic modifications and the non-enzymatic modifications. Now we have to take note that the non-enzymatic modifications are generally classified under degenerative post-translation post PTM, as they actually lead to the different the cessation of the protein function, which are shown your oxidation, glycation, and you know, which that being oxidation being the utilization of reactive oxygen species, and the glycation, which is like the non-enzymatic mediated process of adding sugar groups. So in our presentation, we will be focusing mainly on acetylation as part of the enzymatic modification. Now, the functions of PTM are very varied, and these are the four most studied ones. So the assist in protein folding, an example would be like glycosylation, localization of protein to cellular compartments like your RAF-ER due to N-terminal acetylation, activating or inactivating catalytic activity, which is a very essential uh, process in your signal transduction, which is usually mediated by phosphorylation. And last but not least, one of the most commonly studied PTM would be ubiquitination, which really serves as a marker that target proteins for degradation. So acetylation, what exactly it is? It's actually the process of introducing an acetyl functional group to a compound. And in biological system, the acetylation is actually mediated by uh, a substrate called acetyl coenzyme A. So there are two kinds of protein acetylation: N-terminal acetylation and lysine acetylation. In the N-terminal acetylation, it is one of the most common co-translational covalent modifications of protein in eukaryotes, which constitutes around 85% of human proteins. So on this diagram, you can see that the, the alpha amino group on your N-terminal uh, amino acid, which exists your ammonium ion here, is actually attached to an other to an acetyl group, which is mediated by N-terminal acetyltransferase. So the reaction catalyzed by this actually uses acetyl-CoA as a substrate as a donor, sorry, to transfer acetyl group to your protein, and specifically to the alpha amino group of your first amino acid residue. So diving more into your NATs, this diagram actually shows the different uh, specificities of different enzymes of NATs. So in each the different enzyme complexes, they have specific substrates for amino acid to be uh, N-terminal acetylated. So you may be wondering, if your N-terminal of your protein is actually acetylated, why wouldn't it be the methionine in general? Well, and this is because in some cases for your proteins, the methionine is actually cleaved, and the subsequent amino acid, for example, serine, alanine, or glycine, is actually acetylated, and therefore this actually shows that the different NATs actually has different specific substrates of different amino acids to be uh, to be acetylated at your N terminal. So one of the function of N-terminal acetylation is the most studied one would be regarding protein localization. So what about protein localization with respect to acetylation? So the organization of the membrane traffic actually requires this GTPase, which is R-flight, and this R-flight GTPase actually require N-alpha acetyl group, and therefore acetylation plays a role in your N-terminal for the targeting movement of protein to the Golgi body, and therefore this is actually useful are more important for protein trafficking to steer your protein to the Golgi body. Now, the next example of lysine acetylation is as shown, where the acetyl group is actually transferred to the epsilon amino group of your group lysine residue. So from here, you can see that your epsilon amino group actually contains a positive charge. So the addition of acetyl, the acetyl molecule donated by acetyl-CoA actually neutralizes a positive charge on your lysine by introducing an amide bond, and therefore this actually increases the hydrophobicity of protein, and at the same time removes positive charge on your protein, reducing its ionic strength. So one of the most studied as lysine acetylation in the literature would be that of the histone proteins. So as you can see from the diagram, the acetylation of your histones on a lysine residue actually neutralizes the positive charge of your histones, and the histones are actually responsible to form octamers for nucleosomes, which actually is responsible for the DNA compaction. So therefore, the so in your DNA compaction, when since your DNA is actually negatively charged, the positive charge of the lysine residue of your histones actually creates this ionic bridge between DNA and your histone proteins to compact it. So the acetylation of your lysine as a result will actually remove the positive charge, and this is functionally important in your DNA as the acetylation of your histone proteins actually upregulate gene transcription, and this is actually mediated by histone acetyltransferase. So the reverse of this process, the deacetylation mediated by histone deacetylase, actually aids in the DNA compaction and cell division, and this is most prominent in the prophase 1 of your cell division. However, now the list of functions of acetylation are not exhaustive, but we have listed the ones that are the most extensively studied. So two other examples of acetylation would be that of the p53 tumor suppressor protein, which actually initiates apoptosis under severe DNA damage. So the acetylation of several lysine residues, as shown, actually activates the function of the p53 protein to prevent uh, cancer cell growth. Now another example would be tubulin. So what tubulin actually does is it maintains the structural integrity of cell and also the movement of the organelles. And therefore the acetylation of the residue lysine 40 on alpha tubulin is actually a hallmark of stable microtubules. So this actually uh, ensures that it is not degraded at any circumstance, so that the structural integrity of the cell can be maintained. Now to delve in more specifically into cardiovascular diseases, acetylation actually plays quite an important role in heart failure. In a study done by Fukushima and Lupashix in 2016, they actually found that the accelerated fatty acid beta oxidation rates were actually more prominent in obese hearts, which actually induces cardiac dysfunction. So how does that actually arise? So as you can see from the diagram as shown here, the acetylase there are the enzymes, mitochondrial enzymes involved in the beta oxidation pathway actually has their lysine residues acetylated, and this actually leads to the metabolic change as it increases cardiac fatty acid oxidation rate and also uncouples glycolysis. So the whole metabolic shift of utilizing fatty acids rather than glucose actually is a prominent sign in obese hearts of patients, which actually might lead to a potential risk of cardiac dysfunction. So moving on to the case studies that were presented to us, we actually have done an analysis of the two clinical samples obtained from the aortic tissues of patients suffering from cardiovascular diseases. So as shown as a screenshot on the Excel file here, now uh, we actually arrange the peptide score, expect the e-value, and as well as the setting the parameters of the variable modifications. So in the variable modifications, we actually set uh, in this case study acetylation, and also two more which are actually oxidation and deamination, because these two non-enzymatic processes are commonly found in proteins across the body. And if this one, without factoring these two modifications in, it will lead to actually an inaccuracy in the score, because these processes are very common in almost all proteins. And as such, our variable modifications are these three, which is acetylation, deamination, and oxidation. So this peptide score actually refers to the homology of the peptide with respect to the protein. So uh, we have actually ranked them according to the highest to the lowest, and combining both files together, we actually found the highest score to be 162.22 for 5 samples. So with the high score and also the low e-value, where the e-value actually corresponds to the chance of obtaining a false positive, which is actually the lowest of all, we can actually this actually suggests that acetylation in these 5 samples is actually a very significant PTM. And if we were to look at the description of proteins in these 5 samples, it actually shows uh, sorbin and SH3 domain, sorbin and SH3 containing domain in all these 5 samples, and therefore our analysis would be narrowed down to this protein. So this protein is actually also called SOPS1, where its full name was as mentioned before. So the alternative names to this are actually ponsin, SH3 domain protein 5, SH3, P12, and CCBL associated protein. And the ascension code is actually Q9BX66, and it is actually 1292 amino acids long. Now to show the full amino acid sequence here, we can see that the peptide extracted from here actually lies well between the 361 region, where more specifically the 368 lysine residue was actually acetylated in this peptide sequence in the SOX1 protein. So what exactly is a SOX1 protein? It is actually made up of a conserved sorbin homology domain and three SH3 domains. So they are detected in the skeletal muscle and is also widely expressed in the highest levels in the heart and skeletal muscles. So one of the main functions of this protein, which was very extensively studied in literature, and this actually has found that it actually plays an important role in the tyrosine phosphorylation of CBL, and this actually does so by linking CBL to the insulin receptor, and this actually is required for the insulin stimulated glucose transport, and also it also is involved in the formation of actin stress fibers and also focal adhesions. So with that being said, that's not the only function of the SOX1 protein. As you can see, various uh, there are actually various functions, cell biological processes that are actually associated with SOX1 protein, and more specifically to cardiovascular diseases, we actually narrow it down to the focal adhesion assembly. So it's actually the aggregation and bonding together of a set of components, or rather a complex of intracellular link, signal, link molecule, and structural proteins that actually provides a link between the cellular actin cytoskeleton with the ECM, and it also functions as a locus of signal transduction activity. Now, in a more recent study, it actually shows that pexilin and ponsin, which is an alternative name for SOX1 protein, actually interacts in your costomeres of the muscles. And the key findings of this is that the overexpression of ponsin resulted in the altered muscle cell matrix contact morphology, and this actually implies its involvement in the establishment of mature costomeres. So, what exactly are costomeres? Costomeres are actually functional component of the striated muscle cells, and its functional importance is to basically connect your sarcomere, which is a functional unit of muscle fibers, to the plasma membrane of cardiomyocytes. So so the maturation of costomeres actually facilitates the adhesion of sarcomeres to the plasma membranes of cardiomyocytes, which are your heart cells involved in heart contraction. Now, to zoom in further to sarcomere are actually a basic unit of shredded muscle tissues, and they are it's also composed of long fibrous proteins, which are your myosin and actin filaments, and it's the involvement of its action of sliding your myosin and actin filaments across each other actually leads to your muscle contraction, which actually stimulates heart muscle movement, which is a very vital function of your heart, because you have to pump blood from your heart. So to summarize our findings, uh, this are this is a flow chart of whatever that we have done. So to analyze the clinical sample of our Excel file, we actually targeted the substance protein, and from the substance protein, we actually show the association of the maturation of costomeres with this protein, and this maturation of costomeres actually results in the adhesion of sarcomere to the plasma membrane of your heart muscle cells, which is important because the sarcomere actually leads to cardiac muscle contraction. So this of so this first pathway actually is a conventional function of the substrate protein. Since there were no studies conducted to elucidate the role of acetylation in substrate protein, we actually hypothesized that the acetylation of the lysine residue, specifically 368, uh, a region of your substrate protein, uh, actually actually affects the maturation of costomeres, which impairs the adhesion of which no sorry which actually inhibits the adhesion of sarcomeres to your plasma membrane of cardiomyocytes. So from there, there will not be any cardiac muscle contraction, and the impairment of cardiac muscle contraction actually leads to an illness called cardiomyopathy, where it is characterized by the weakness of your heart muscle cells. So from here, we hypothesize the potential role of acetylation of the SOX1 protein uh, in relation to cardiomyopathy. Okay, so this is our hypothesized workflow to actually compare the amount of acetylated SOX1 protein in patients suffering from cardiovascular diseases versus the SOX1 protein from healthy individuals. So at the first step, we actually label the SOX1 protein with isobaric tag. So this will be identified during mass spec in the later steps. So from there, after labeling of your SOX1 protein with the isobaric tag, specifically to the acetylated region of your SOX1, we propose the preliminary separation of the SOX1 protein from tissue sample. So this will be done by STS page, and from we can actually isolate out the bands of SOX1 protein because we actually know the exact molecular weight as proven by theoretical findings. So from there, we actually do an indel digestion with trypsin enzymes in order to get peptide fragments of your SOX1 protein, and this will and the uh, trypsinated peptides actually we will use that to purify it under fractionation using HPLC in order to extract to get to obtain a better quality and yield of our desired SOX1 protein. And our last step would be to run your quantitative proteomics, which would be a Molotov MS. And the reason why we do so is because we actually have isolated out the protein of interest. So therefore, we would choose Molotov MS because it's actually uh, it's more efficient, and it will take less time to obtain our readings, yeah, in general. So from there, we actually compare the varying peaks of intensities with the various reporter texts. So we would actually compare, because since we are hypothesizing that the iTrade label actually uh, is tagged to the acetylated SOX1 protein. So if we check if there is a difference between the intensities which actually correspond to the protein abundance in the samples. So if your acetylated SOX1 protein in your patients is actually significantly higher than your SOX1 of your healthy individuals, this actually suggests that the acetylated SOX1 protein may actually play a role in the onset of cardiomyopathy. However, if there's no significant difference between the intensities of the peak of the eye tract label in both SOX1 protein samples, this actually would suggest that your acetylation PTM may not even play a role in the onset of cardiomyopathy. And from that, we come to the end of our presentation.